Okay, welcome everybody to another session of the Quantum Computing Talks hosted at the University of Porto. Today I have the pleasure to introduce Dr. Shahar Keinan, who is the co-founder and CEO of Polaris Quantum Biotech. She has over 20 years of extensive experience in the field of computational and theoretical chemistry and published over 50 peer-reviewed manuscripts in the fields of in silico drug design and discovery, as well as molecular materials design and computational methods development. Dr. Keinan has a PhD in theoretical chemistry and was previously the CEO of Cloud Pharmaceutical. And today, essentially, she's going to tell us, we hope, all about her work on Polarisqb work on drug discovery. Thank you very much for taking the time to talk to us. Please take it away.
Sure, and thank you guys for inviting me over. I always enjoy these kind of things. I think part of my job and part of every scientist's job is, of course, telling others what we do. I'm not going to say the words of educating the public, but definitely sharing with how some of those technologies can work and can improve the lives of people, of patients, and of, again, the public and patients. So what Polaris is doing is we are doing computational drug discovery, computational drug design much faster than anybody else. And we can do that because we are using quantum computing. Basically, what we do is for a specific disease, for a specific protein that is relevant for that disease, for a specific binding pocket in that protein, in that disease, we build very large virtual chemical space, billions of molecules, and we use quantum computing to search it, to find the best molecules inside that large chemical space. And I'm going to show you how we're doing it. We published a bit about that, and if anybody has questions, I'll be happy to answer those. So why us? We have the first drug discovery platform which is built on a quantum computer. So we have this platform working right now. And what we do is in a molecular space of billions of molecules that are possible, we find the right one. I'm going to start with a bit of a tangent, which is what kind of quantum computers are available right now and which one we are using. And this is really a very limited view. And I basically took a whole bunch of computers, quantum computers, and I bunched them together, right? So it's, some people call them gate, or these are the gate quantum computers, but also the ion traps, atom traps, all of those kind of quantum computers that can solve all quantum computing problems. And they can do everything from annealing, but also solving the Schrodinger equation, for example. So that's gate computers. This is what IBM and Google and Rigetti and IonQ have. The problem with those systems is that they're still quite small. So I think I need to update my slide here. It's not 32, it's 50 something qubits that is available. But the problem is that these is still a very small system. And there are interesting developments there, but it is not ready yet for a commercial use, because what we need is to work with 2,000 to 3,000 qubits. And in order to do that, we are working with quantum annealers. So this is Fujitsu or D-Wave. Fujitsu is not a fully quantum system, it's a digital annealer, but D-Wave is a quantum annealer. And D-Wave, their current system, it's called Advantage. It has 5,000 qubits on it. The limitation of that system is that it can only solve optimization problems. So it is an annealer. What is an annealer? An annealer is a system that can use difference of temperature to stabilize a quantum system. Now, interestingly, annealing is a process that humans have been using for a millennia. maybe even more than that. So whenever you prepare iron, for example, what you do is you heat the metal and then you cool it very fast. That's an annealing system. So you heat the system, it goes out of its local minima, and then you cool it and it goes back to the global minima of your system. And I'm showing you here, and this is taken, like a lot of the background stuff that I have is taken from D-Wave. So I'm showing you here is how we should think of annealing inside the quantum computing. So you start from your system at a superposition state, right? All of your states are in a superposition. And when you start separating your system, you heat the system and you cool it, you separate it into the 0 and the 1 ground state. However, you can start applying magnetic field, which will make one state more stable than the other. Okay. So then the lower state, for example, 1 here, has a higher probability. But if you are at the 0 state, you will not be able to move from the 0 to the 1 state unless you heat your system. You heat the system and then you cool it. When you cool it down, it will go down to the low probability, to the high probability or lower energy state. Now, this is just for a 1 qubit system. Of course, if you have 2 qubits, your system is double the size. If you have, of course, 3 qubits, then your system is again doubling in size. And you can see here the same way you have you're using magnetic fields to give or include your constraints or your preference for some states. And you that way you are finding the low energy state, which is the higher probability state or the answer to your questions. The D-Wave system have been used before, and I'm going to show here 2 examples of what is it good for. So what kind of problems can we solve using an optimizer? Because that's the annealer, it is an optimizer. One of the problems that they looked at was, how do you do a waste collection route? So you have the cities change every day, where there's more garbage or less garbage also change. And so on a daily basis, you need to optimize your route. This is something that, and you have a limit number of trucks, a limit time frame that you want to do that. And that's one of their optimization problems, where they can put all of that into a single matrix, and the number of routes that are possible is, of course, exponentially large, because the trucks can go throughout the whole city. And this is, and you can think of it as a version of the traveling salesman problem that is solved very efficiently on an annealing system. A different problem that you can solve is personnel management. And this is with a company, a large Japanese company. And you can think that when COVID started, so one of the first things that happened is that the number of people that can be in a certain location, in a certain office, of course, went to about half the number of people. But they always needed to have some people with some properties or some definitions. So you always need to have a reception person. You always need to have a nurse in the same building, but the number of people have to be half, and then you have to make sure that people themselves have maybe this day they cannot come to work, this day they can. So that kind of a personnel management is again a large optimization problem that they implemented on D-Wave. And the algorithm or the kind of problem that they used here is not traveling salesmen, it is the backpack problem. What do you feel inside a backpack? So these are the kind of problems that we are using, or the kind of algorithms that we can use to do that. Why do we use that? quantum computing, we can, I want to make sure that everybody understands that we can run those problems on a Google Cloud or Amazon Cloud. So we can run it on a silicon-based computer. We will probably not be able to do that when instead of having 1 billion molecules, we're looking at a library of 100 billion molecules. It does not make sense at that time. But right now we can do that. But we are using quantum computing. So why? The first is the cost and the time. So it cost us about $40 to run on a library. These things are counted by, tenth of a second. That's how long it takes to run on our billion something molecules. To run this on GCP, and this is a brute force, it goes about $40,000. So for the same size of the library. Of course, if we run this, if we increase the size of the library by a factor of 10, so instead of doing 1 billion molecules, we will do 10 billion molecules. When we run it on Google, it will cost us $400,000. When we run it on D-Wave, because the size of the efficiency of quantum computing, it will be a bit more than 40. It will not even be $400. It will be more like $80. So quantum computing, we can do bigger libraries and it scales much better. Also, we are interested in optimization problems and other optimization problems. Quantum computing is the way forward to solve those kind of problems. It's more efficient. The algorithms are running way faster and make more sense to run them on a quantum computer. And of course, we are getting ready for quantum computing. So the process that I'm describing here now, just about half the process is run on a quantum computer, and then we take the results of that part and move it to a silicon-based computer. What we want to do is to run everything on a quantum computer. So we are getting there. Not yet, as I said, gate computers is still not there, but we're waiting for it. I've went through that already. Why quantum computing? When we look at over 1 billion molecules in an efficient way, this is the only technology that can do that. There are other methods that people are using, but they are usually methods where you don't look at all of your solutions. So something like a branch and bound algorithm, where you say, okay, I'm going in that direction. That direction, the first ones that I'm calculating in that direction doesn't seem so good, so I'm bounding that branch. I'm not going to go into that area. Okay, so you can do that on very large libraries, but that means that you're missing some of your answers. The only technology where you really look at each and every one of your solutions, each and every molecules in that chemical space, is quantum computing. Another interesting thing here is that we're doing a multi-object optimization, really a multi-object optimization. We don't decide a priori that this property is more important than this property. We're just looking for the molecules that have both of these properties. And that's something that's not easy to implement on a regular computer. So let me tell you a bit about the process that we are using. And the problem that we are targeting is that currently, when people are designing molecules on the computer for drug design, drug development, people know how to do that. So this is something that the industry has solved, how to design small molecules that will be good binders for a certain protein pocket and a certain disease. The problem is the scale of that. People can do that for a small number of molecules, 100 molecules, 200 molecules, maybe 400 molecules. So you do the design on the computer. And when you finish with that design, you choose 20 to 50 molecules. You synthesize those molecules, which is a expensive, time-consuming, and then you go and you measure the activity of those molecules. And then you learn something. You definitely learn something. And you go back to your drawing board and do some more designs on the computer. And that's because you can only look at a certain number of molecules every time. This process of design, synthesis, and measurement is an iterative process that the industry does it 3-4 times sometimes in order to find the best molecules. It takes a couple of years and it's a quite expensive process. What we're saying is, instead of looking at a couple of thousand molecules every time, look at a much bigger chemical space. So we are looking at billions of molecules and evaluating all of those molecules at the same time, evaluating all of those molecules at the same time to find the best molecules there. Sorry about that. So that's, again, instead of looking at parts of chemical space, we're looking at all of it together. And then you don't need to do those multiple cycles of calculations, synthesis, and measurements. You do this just once. Our program, our platform is called Tachyon. It is an automated, it's an agile platform. And because the way we built it, it supports both scale up, but also parallel projects. So we can do multiple projects at the same time. We start from collecting information, bioinformatics, and choosing a biological target, a protein that we want to work on. We are using there some NLP, some machine learning to find the best biological target for a specific disease, and also for defining the properties that we are interested for that specific protein target and disease profile. The next step is we're building a molecular library where we build a solution space of billions of molecules. And then we use quantum computing and some high accuracy calculation as well to find the right molecules. We do synthesis, we do measurements, and then we can either sell or license that as a pharmaceutical asset. We have, I'm going to discuss here now a white paper that we published specifically on that process. Here is the address for that white paper if anybody is interested in that. The white paper was published with the Fujitsu digital annealer team. They're located in London. The computer is located in Japan. And we, when we set to do that, that was over 2 years ago, we chose the dengue fever as our target. I'm guessing that if we would have started that project 3 months later, we would have chosen a different virus, quite sure about that. But when we started that project, dengue seemed like a major health concern. It is a mosquito-borne tropical disease. It's caused by the dengue virus and transmitted by mosquitoes. And it is a nasty disease. High fever, muscle and joint pains, skin rashes. People really suffer throughout the disease. However, but usually they recover. In a small proportion of the populations, the disease can develop into severe dengue, which is a hemorrhagic fever, which means that most people do not survive that, the hemorrhagic fever. There is no drug for dengue right now. There was a lot of effort to develop a vaccine, but the current vaccine is only recommended for a small number of patients, and that vaccine activity almost stopped completely. There's some interesting work done on that, especially in the Philippines, if anybody is interested in that. So after we decide that we are interested in a specific disease, the dengue fever, the first part is, of course, to choose a biological target. And we chose this target, Dengue RNA-dependent-RNA polymerase. And one of the reasons that we chose it is because Novartis, which is a large pharma company, worked on that specific target. And they have done a series of 3 publications, each one that took about a year of work. So they've done the same process that I described to you before. They've done design on the computer, they went, they synthesized, they measured, they learned, they went back to the drawing board. And each one of those cycles of design, synthesis, measurement is a paper. So we had a history of the work that they've done that we can then take a look at it and say, okay, can we reproduce that and maybe shorten their time scale? So they started with something that is called the fragment campaign. So they looked at the small pieces of molecules and where do they bind in this specific protein, and they found a pocket. And a year later, after they've done the first and the second design work, the first 2 manuscripts that they published, and they found that they can add a hinge region for their molecules. And that hinge region substitute a water molecule. So basically their molecule is bigger and is removed a water molecule from the binding pocket. And that increased their activity dramatically. And they published, so this, there were 3 papers that describe the work that they've done. So this is a good preliminary work that we can compare to. And this is the binding pocket that we were interested in. After we after we chose a target, the next phase is to build a molecular library. And we can do this in different ways, but the way we built it here is by saying, okay, if you look at the bottom part of the slide, this is for a specific binding pocket, specific protein environment, and a set of physicochemical constraints, so properties of your molecules. We analyze the pocket of the protein and build a library that fit into that pocket. This is a combinatorial library. So we choose a central scaffold and R groups around that scaffold, and we can move the scaffolds, we can move the R groups, and that's how we built a very large library. We make sure that the molecules in that library are synthetically accessible. So these molecules have to be tested to make sure that we can synthesize them later on. And when we do that library, one of the things that we have to make sure about is that we can translate that library into a quantum computing. So how do you formulate the molecular properties as a QUBOs, or quadratic unconstrained binary optimization object, which is polynomial, which is what we optimize at the end of the day. One of the things that we care about is how do these molecules fit into that pocket. So I'm showing you guys on the right-hand side of this slide. For example, we are interested in a certain size of molecules, right? So that they will fit into the pocket, and certain properties, hydrogen bond donor, hydrogen bond acceptors, again, fitting into that specific pocket. And the optimization goal of all of these QUBOs polynomial is to find all the molecules that in that specific chemical library that match those criteria, these imposed criteria, both physicochemical constraints and structural constraints. And that's from a library of billion molecules. So then after we built the library, we built the constraints, we ran it on a quantum computing. We used the Fujitsu digital annealer to do that. And we started here from 1.3 billion molecules. Here is a fast process, or fast overview of how you do this on a digital annealer. It's a bit different than a quantum annealer. because again, it's not a quantum system. It is a silicon-based system. And they start by random initialization, and then allow variation there. So it's more of a, it is an annealing process, but I think it is less clean than an annealing process on a quantum computer. Was there a question?
Yeah, yes. So, by the way, it's very timely, this interruption, because we had a question by Alex Feldman, which is precisely about the mapping of your optimization problem as a QUBO, right? And so, in particular, if you're using D-Wave, the question then becomes, what if your QUBO does not fit the chip, that is the real hardware? How do you split it into smaller QUBOs? If that is always possible, I suspect there might be instances in which that is not trivial, or if you want to break them, you generate a handful of additional QUBOs. I don't know if that is what happens, right? So that was the question.
So interestingly, Fujitsu is almost a planar kind of system. So all all chips or in the Fujitsu system have the same connectivity with one another. But the D-Wave system is more of like a tree. So each QUBOs has, or each qubit has connectivity to close number of other qubits around it. Okay, so on the Fujitsu annealer, you don't need to think about those kind of questions. You hardly reach the time where you fill the whole system. But when we're running on D-Wave, we do have to think about those questions. Let me show you here. So here is, and this is how we implement a different system. Okay, not the white paper, a different system that we worked on, and how we implemented it on D-Wave system. Okay, because the D-Wave system again is a different architecture than the Fujitsu system. So one of the interesting things that we see is that our R groups, okay, so we have a scaffold and R groups. And because this is chemistry, you can have a single solution for each R group. So if this is R1, we may have 60 possibilities for R1, but only one of them can be chosen. But so there is a constraint that is choose one out of R1. So all of R1 are connected with the same constraint. So here, for example, the left-hand red blob is all of the R5, you can only choose one of them. So then when you implement it inside D-Wave, you have to think about these things. You have to make sure that all of your constraints about R5 are on the same area of the chip itself. So you cannot have, R5, one group here and one group over there, because that's extremely non-efficient.
Gotcha. Okay.
Okay. So that's one thing. The second thing is you can reach the time where even that is not enough. And then we do something that is similar to divide and conquer. Okay, so we take our library and we divide it into 4 quadrants, and then you do 2 quadrants every time. Okay, so you run half the library 4 times. So you choose all possible combination and you look at the results of that. So there are ways of dividing the library in certain ways.
Okay, all right.
Okay.
Let's. I suspect that you
I don't have that much more. Yeah,
right.
give me a couple more minutes.
Yeah,
couple more minutes.
I actually have a couple more questions along those lines.
Yes.
But let's go through your presentation. Okay. Thank you.
So if this is the weighted penalty graph, so this is if two groups are connected with red, that means that they have a constraint. If 2 groups are connected with green, that means that they have an incentive there. And I'm not showing you the whole graph. Okay, it's much more complicated than that. I'm just showing you parts of the graph. And the outcome is a single, this is the solution graph. So we chose R22, R4, and R43, for example. So this is the green line here is just the solution based on the previous graph. Okay. And I'm going back now to results from the Fujitsu digital annealer, and we show that we do find better molecules, and we are comparing here a random or an anneal result. And you can see on the right-hand side that there are over 2 orders of magnitude difference between the anneal results versus the random results. And so we do find the best molecules. The molecules that we find have good drug-like properties. So here is the molecular weight or log P. So we do see that we are finding good molecules. This is a summary of the work that we've done. This is a published work. We designed a very large 1.3 billion molecules. We used the digital annealer to start from billions and go to about 3,000 molecular leads molecules, and that took less than 5 minutes. That specific run on Fujitsu system. I told you before that it took Novartis team 3 years to find that hinge molecules. About half the molecules that we found in this run included that hinge region. So instead of doing it in 3 years, they could have done it in 3 months or 4 months. That's how long it took. And we also identified some novel molecules that are being synthesized right now. And if anybody is interested in what we're doing today, we have a collaboration. This was just published 2 weeks ago, a collaboration and where we're looking at a cancer targets with a company called Foremost that is located in Cambridge in England, if anybody is interested in that. We start from a target and a profile. It takes us 3 to 4 months. And at the end, we have 20 to 50 molecules candidates. And this is, I want to say here something that is, we are one of the first companies that is using quantum computing and selling platform using a quantum computing to any kind of a commercial activity, not as a one-off, right, but as a platform. So this is the kind of PR that we're doing, asking pharma companies, right? And for pharma companies, they don't care if we're using quantum computing or not using quantum computing. What they care about is how fast and how good are our molecules. And this is where quantum computing, I think, can really shine by solving problems today faster and more efficient. Thank you, and I'll be happy to answer questions.
Okay, thank you very much. I'm sorry we don't have here an applause kind of thing, but I really enjoyed your presentation. I had a couple of questions, but I would like to give the opportunity and the floor to other people. So let's see if Rui has a couple of other people from the YouTube channels.
Not yet from YouTube, but I keep an eye on it. Yeah.
Oh, okay, thank you. So I would like to go back to your discussion on the algorithmic aspects. Obviously, this is clearly a person with a computer science background. You mentioned at a given point, divide and conquer. And from my theory of algorithms days, the divide and conquer works very well, in fact, optimally, for certain class of problems, the so-called matroids and whatnot. So were you, I mean, do you, what I had in mind saying is that when you take that approach, typically it's because the space is so huge that you're trying to limit it, the search space. I mean, have you felt, I mean, you guys at Polaris felt that you're missing some opportunities for optimization, or you think that this is very suitable at certain level to be partitioned, because the likelihood of being a molecule that does not fit your partition is so low that that might even be the case, right? So I'm wondering what you feel about this. I know that you did this for a practical need, I suspect, right?
Yes. Okay. So far, we didn't need to do that, because although our libraries are big, there are billions of molecules, we are still using we are still not reaching the limit of the quantum computer, of D-wave advantage. But this is something that we are thinking of. I'll tell you, we want to increase the size of the library and increase the number of properties that we are using to constrain. And I believe that give us a couple more months, we will reach that limit. Now, D-waves are saying that they are going to increase the size of their computer as well. So it's a competition, right? It's a race.
Yes.
But if we don't have to use divide and conquer, we will prefer not to use it.
Right. So, okay. So the other question that comes to mind is, can you combine this as a hybrid approach to computing, where you have traditional computing, even high performance computing? I know it's expensive to eliminate some candidates. So essentially constrain the search space or reduce it substantially, so that you basically are always one step ahead of the game in terms of the quantum machine. You don't have to wait for D-wave or Fujitsu to come up with a bigger machine when you really need it. So I'm wondering if the type of problems you're looking to are amenable to these pruning early on of things that this is impossible, or the likelihood is so low that you essentially do a branch and bound followed by quantum. I don't know if this is something that makes sense.
So we are using high throughput computing, and we are using it in different steps. So the first thing we do is we built a very large library, and we use quantum computing to go from billions and to 100,000 molecules. That's usually the size of the number of molecules that we find. Now, from those 100,000, to choose the right 100 for synthesis, this is usually where we use either cloud computing or high throughput computing, and we are using some of the other computational chemistry methods. We don't do branch and bound at that point. We do some calculation on the properties of the molecules to make sure they are where we want them.
So it's actually it's actually the other way around that I was thinking. So you're actually using quantum to eliminate a lot of this, and then just focus on the other. I really want to be sure that this molecule is right. Okay. All right. So there was a question here. I don't know if you can understand what this person is asking. It's he or she is saying that he's very curious about the approach used to construct the graph shown with red and green edges, and is asking if you can explain it in a bit more detail.
Sure. So this is a way for us to visualize the library and the constraints that we are bringing in. Okay, so because the library has a couple of hundreds of components, and there is some, we don't want all of our the members of R1. They have a constraint that we only choose one. That's one constraint. But we also have the constraints that, for example, number of hydrogen bonds in R1 and R2 needs to be below 5. Okay, so that's another constraint that we can have. So we have different kind of constraints and different connectivities that between those R groups. So we want to do that visually, because it's difficult to see that in a code. So that's why we moved into that visualizer. And that visualizer basically plots all the components of the library and connect components of the library. Now, that components, if the connection is a constraint, it's green, it's a red, and if it's a something that we want to enhance, then it's green. So that's how this is translated. It doesn't show the strength of it, and I've removed some of them in order to make it more readable. Okay, the graph itself is quite busy.
Okay.
Yeah. Okay. Perfect. Quite clear. And so that's part of the process. Sorry if I unmuted, but I was asked to.
Yeah.
Yes.
And anyway, so that's part of the process that belongs to the extraction of the library of molecules, of billion molecules. Yeah. Okay. So my question is more about how these billions of molecules are like scored in a way that the quantum annealer can manage to like find the solution. I mean, I understand the property of the quantum annealer that finds the minima, but I'm not quite sure of how it becomes an optimization problem. That's the question I'm looking for, the answer I'm looking for. Thank you very much anyway.
Sure. Let's say, and this is, I'm giving you a oversimplified example, okay? Let's assume that I'm interested in a molecule that has a molecular weight. So the weight of the molecule is below a certain number, 500. So each of the R groups has a different molecular weight. And what I'm interested is in a combination of one possibility for each R group, that the total of all of these will be below 500. Okay, now I'm not interested in just one property. I'm interested in multiple properties, and some of them are not just the properties of a single R group, but they are a binary. So we need to include interactions between two groups, and there are others. But this is how it is. So think of this as translating it to the backpack problem, right? The backpack problem says you have a backpack, and you can put items inside the backpack. Each of these items have a weight and a price. And you're interested in a combination of all the items that will give you the highest price and the lowest weight. So it's the same thing here, but we are using chemical properties instead of weight and price. We're doing more than just two properties, and we are doing a very large solution space. Okay, so this is an optimization problem. It is not, that's what it is, right? You are optimizing the best R groups in it.
Yeah. Okay, actually, as a segue to that, you mentioned in your flow, let's say, of drug discovery, that at a given point there's a synthesis, a test, and then you mentioned stability tests. Is that already folded in these constraints, or at this point in that flow, you basically hope that everything that meets these constraints is stable and you check them later? So in other words, could you actually a priori prune some molecules that are clearly unstable or extremely hard to synthesize? I don't know if this is feasible.
So the synthetic accessibility is something that we do include in the quantum computing step.
Right.
And I think we discussed this during the white paper. So you can see it in the white paper, how we do that. There are some groups. So each R group separately, we make sure that it's stable. But sometimes when you put two R groups near one near the other, that's not stable. Or when you put them three R groups, and we can do binary, we cannot still do the interactions between three groups. So that's something we calculate later on by the 100,000 to a couple of dozens of molecules.
Okay, good. So there's another quick question from the YouTube. Are there any plans or use cases to explore parallel processing across either existing HPC or quantum processing units? You may not be there yet, but
I wish there was. Okay. We are using HPCs all the time for different stages,
right?
But for other quantum computings, QPUs, we are trying to work with other companies, of course. They're just not there yet. it's the size of the system is still quite small. Sadly, I really want to be able to do that because one of our last steps is a quantum chemistry calculation where we are solving the Schrödinger equation. If we will be able to run all of this on a quantum computer, we will be instead of running it on a quantum computer, taking the outputs, moving it to Google, to GCP, and then running it there. If we can run everything on a quantum computer, this would make our life very efficient and very happy. Yep.
I also have a short question, may I?
Yeah, sure.
Well, thank you, Shahar, for your nice talk on this pioneering quantum computing potential application.
No. Not potential. We
It's
are
real.
running it today. It's real. We are solving that. We
Are
are
you
running
using
it today.
digital and not quantum annealing?
We are using we are using D-Wave. We're using
The real
quantum
iron.
annealer, the real iron.
Well, but my question is just about a small detail. I'm sorry about that.
Yeah, sure, please.
But it's not the case that in the annealing process, you don't cool the metal or whatever function parameter, for example, very fast, but on the contrary, very slow by making small temperature variations.
I thought when
all about
when,
annealing is to cool very slow, not very fast.
Okay.
I thought there could be. I think, but
We have
I'll tell you.
Yeah, I'm
When
not
I-
sure
when we
either
do-
exactly how
when
the
we do
cooling process is.
simulation, when we do simulation, we control the cooling speed,
Parameters.
definitely. Yes, but when people used to make swords, right? Swords for
Yes.
fight, right? They hit the sword and then they put it into cold water. So that's a very fast cooling.
But annealing is just about making temperature variations very slowly.
Yeah,
I'll
I
check.
suspect
I
that,
will
that
check. I will
the
check.
speed, I think the speed is possibly connected to the not trying to get caught in local minima. And so maybe you are exploring a little bit the speed so that you see whether or not you get trapped. Maybe there's something to that. Okay. So
I think, Emmanuel, I think you're right here that we need to do it. We avoid the noise and being trapped at local minima.
Yeah. So there was a, I mean, Eugenia, I don't know if you.
That's okay, thank you.
That's okay. Okay, if this answers your question or at least clarifies. Okay, we have another question here. I'm not understand exactly what the person is asking, but maybe you do, and basically says the following: we have reached the level in quantum computing where we can optimize properties such as Lipinski rule 5 across billions of molecules.
Yep.
And could you elaborate how you encode R groups into qubits? I don't know.
So I agree that we can do. We are doing Lipinski rule of 5. I think that's the basic part of it. We are trying to do more than that, much more than just Lipinski rule of 5. We try to design libraries that have the Lipinski rule of 5 built into those libraries, right? So we can make sure that the molecules, the library itself follow those rules. I'm not sure I understand the second questions, but what we do is each of these R groups have several values, right? Again, I'm going to the molecular weight. It will have a certain molecular weight. And then you take that molecular weight, that would be your qubit value. And of course, it's a multi-object optimization, right? So you need to optimize rule of 5, molecular weight, and only choosing one R group versus the other. I'm not sure if I answered the question.
Oh, it's perfect. Thank you. Thank you, Shahar.
Sure.
So Rui mentioned that there's another question, but sadly I cannot see it. Rui, can you?
Yes. So I have just unmuted Anuj, but the question is, are there any plans or use cases to explore parallel processing across existing HPC and QPUs? Anuj, feel free. Oh, you have asked that.
Yeah, we asked that.
Oh, I'm sorry, I missed it.
Okay, all right. Okay. Good. I don't know if there's any other questions. I do not have any other questions at this point. I'm very interested, by the way. I mean, another quote unquote simple question, maybe it's not that simple, is why not Ebola virus at this point? Is it radically different than dengue? Because there's also no cure for that either, right?
Yeah. So this is almost a very different question, because that's the question of a commercial application. So well, yes. As a company, we are doing two kinds of projects. The first one is when we have customers that come to us and say, design something for us. And the second one is where we have targets that we are interested in. These are targets that we need to be able to sell at the end. License or sell. We have one which is dengue, which is an orphan disease. And the other targets that we're looking at are more of a cancer or other kind of diseases where we think we will be able to sell those assets easily. Ebola, I think right now there's still not a lot of market for it. And I would like to see how our dengue results are accepted.
First.
The whole area of drug design, drug discovery went through an upheaval, of course, with COVID. And right now, it looks like the whole industry is interested in either COVID or diseases where you have enough information and the patient population is strong enough. So Ebola is not there. There's another questions of testing, and Ebola is difficult to test. So even when we have good molecules, Ebola testing on Ebola virus is very difficult, right? As you can imagine, the level of protection that the people who are doing the testing need. Very difficult to test.
So it's very difficult to isolate them and test them in the lab, right? Okay.
Yeah.
All right. Yep. Okay. Thank you. There's actually one more question here, and I think that we should wrap it up after this, which is about the positioning. And whether or not, do you also find the pocket with quantum annealing, or do you test the billions of molecules already positioned?
So yeah, we're not finding the pocket with quantum computing. With quantum computer, we think we have some ideas about how to do that, but we're not there yet. We start from a known pocket, and the molecules, we know how they bind in the pocket. We assume we know how they bind in the pocket.
Right, and for this you use the quote-unquote traditional high-performance computing method.
Yep.
Okay.
Yep.
All right. Okay. Good. This was very interesting and very enlightening, and this is a real application. I'm very happy. And well, thank you once again for taking the time.
Just one last question,
one last question
from YouTube. So the question is about positioning, and it is: do you also find the pocket with quantum annealing, or you test the billions of molecules already positioned?
Yeah, we just did that, Rui. We are out of sync here.
I'm sorry.
We're out of sync. I suspect that you're unmuting and checking the YouTube.
Yeah, that's true.
Gotcha.
Moving from one place to the other. I'm sorry.
Yeah, no worries.
No worries.
Okay. Thank you very much, Shahar, for this great presentation. Really enjoyed it. And for this insight on how to use quantum computing. This seems to be a really very prolific and great application of this technology. And let's hope that your company does well, because that will be, a good sign that we're going to have better medicine and drugs ahead of us. Okay, thank you very much.
Thank you guys for your time. And I see here people are interested in contacting me. You have my my email in the presentation, and of course you can always contact me through LinkedIn as well.
Okay.
And I'm looking forward to it.